It is Thursday, August 27th, and you're watching the Biotech Voyager. It's the show that covers what's next in biotech. We are mapping the early stage signals before they're big news everywhere else. Jeff, it's going to be a great show today.
Yes, it is indeed.
Right
It
on.
is indeed.
Yes,
Lots of good things to talk about.
Indeed. We've got a special guest coming up at 11:30, the CSO of Camp4 Therapeutics. Really excited to get into them, Jeff. A modality that I feel like we probably haven't spent a whole lot of time on so far on the Biotech Voyager, which is this whole idea of ASOs and how we can start using those for all sorts of different therapeutic applications. So excited to get into that conversation later in the show.
Yeah, antisense oligonucleotides.
Yes.
I'm gonna want to ask him about what is that company that Jennifer Doudna is involved with that I always ask? Scribe. I always ask, what is the benefit of doing like an epigenetic gene suppression when you could just do ASO? So I'm wondering if he has any kind of insight being an ASO guy and being the chief scientific officer. He probably knows more about it than I do.
That could be.
I had to imagine.
Nice. Excellent. All right. So first up, as always, ladies and gentlemen, we'll take a look at the early stage leaderboard. But before we do that, right, Jeff, we should just look at anything. Well, I guess we can start there. Let's go for it.
Yeah. So you know what I should do instead of going to the leaderboard right away is go to the signals and get like the the most recent signals, because this might actually be a better way of looking at like what's happening right now, right? Sometimes you get this huge signal and a company like Kinexus has been #1 for a week, right? But here's some stuff that's been happening like recently. So Hemologics, we're going to go through their CAR T-cell dosed their first patient. Really cool stuff. I think it's in multiple myeloma. You're covering that one later.
I am.
The FDA grants pediatric disease designation for, what is CMT? Dang, I should know what that is. To Revere, you're also covering this one,
yes,
right?
It's a very rare neurological disease, so it's okay if you don't know it.
I was trying to think of like some C malignant T-cell lymphoma. Like, I was like, I don't know what this is. Then we're also going to go through some stuff regarding Tregs, and this might be one of the coolest company names plus logo combination that I've ever seen in my life. T-Rex Bio, and their logo is like a T-Rex eyeball. It's like a biotech company. Super cool. T-Rex, a play on T-Regs. Right. So they got some cool phase 1 top line results from their clinical trial, and then we're going to have on Camp4 Therapeutics, and we're going to talk about an AI-based antibody discovery competition between about 30 companies that was just published in Nature Biotechnology yesterday.
And
so lots of good stuff. And the winner is humanity. The loser seems to be computers. But the winner of the competition, technically speaking, was Aurica Biotechnologies.
Nice.
Which I think they're a Chinese company. Yeah. So we're going to get into all that stuff.
Excellent. Very good. But before we get to all of that, ladies and gentlemen, let me tell you about Touchy Bio. It's all about who you know, but now you can catch prospects the moment they're ready. Deep company research, live signals from your socials, verified contacts, and a whole lot more. BD built for biotech. That's Touchy.bio. All right, back to the leaderboard. So yeah, let's just look at the very top here, Jeff.
Yeah. So the #1 company is still Kinexus. Pretzel Therapeutics, this also really cool company, IND cleared for mitochondrial disease. Their website, Ben, is so cool. Look at their, I don't know why, I just like see that and I'm like, that's interesting.
that's
my initial reaction.
That is
to
a choice. That's interesting.
Yeah, that is a choice.
I mean, you know that it's not made by AI.
Well. Still potentially could be. But yeah, Pretzel Therapeutics shot way up. I put a lot of emphasis on IND clearing, and I'm wondering if it was their first one. I'm going to guess that it was their first IND clearance, so they're now officially a clinical company. Pretzel Therapeutics, congratulations to Pretzel. Who else we got in here? Let's see what Leeds Biolabs. I think that isn't Leeds Biolabs like a discovery platform company? more than
I'm
a
not sure.
company. Well, anyways, this was a really nice, cool thing to see. Degrader antibody conjugates.
Ooh,
Antibody drug conjugates,
I certainly do.
protein degraders. They've had a baby. Antibody mediated degrader delivery.
Okay. Isn't this technically called a DAC, a degrader antibody conjugate?
It's a DAC.
It's a
Yeah.
DAC.
We covered this company, it looks like probably back at the end of April here. They had some preclinical data, but now they are officially clinical. First
Right on.
IND cleared. Yep. So, they raised $100 million in a Series C back in December of last year. They went on a little bit of a touring spree in April, touting all of their IND filing stuff and preclinical data. And then bink, what was this? This was earlier this week. They were clear, or maybe late last week. They had their IND cleared for hematological malignancies. And I wonder if this, here it is. So CD123 targeting antibody conjugate to a GSPT1 protein degrader payload. So it's going to target CD123 to positive cells to degrade GSPT1. And I think CD123 is I don't know. It's obviously on myeloid cells. I cheated because it's for AML. I thought it may have been a T cell marker, but it's a myeloid cell marker. GSPT1 must be some kind of oncogenic driver of this myeloid malignancy. So you can deliver the degrader to the myeloid cells, degrade GSPT1, and cause cell death. So they're now in the clinic. Really cool company out of Lexington, and South Korea. Yeah. Super cool. Oh, and they're public on the Korean NASDAQ. Super cool.
Perfect. I wonder how much their stock has jumped based off of this. Can we go look at that quick?
Ooh. I don't know. Can I? I don't know how to do that. Let's see. KOSDAQ.
No, if you just put in Aurum Therapeutics stock, I'm pretty sure.
Well, I have their ticker.
Oh, okay. Even better.
Let's see. Boop. It went down.
Well, that's today. How long has this been going on?
So the IND was cleared, I think, like last, the 23rd. So whatever day that was.
Okay. Well, you're looking at the last day.
Oh, here we go. What's this? 21st.
Last 5 days.
There you go. It went way up.
Nice. All right.
There.
They got a nice bump.
Right, and then it kept going up, and then I guess it has gone up since then. Yeah, whatever. Yes. Anyways, that is interesting. We should probably, honestly, I've been hesitant. Do you invest in stocks, Ben?
I mostly stick to indexes where I can.
You know what we should do?
Because I'm dumb.
Yeah, I never really understood exactly how it worked. It probably isn't super complicated, but I just haven't taken the time. It would be interesting to see if there's any kind of leverage with the Biotech Voyager, because we could have invested in that. I mean, it's going to go up, right? So we definitely could have, but I think that it gets complicated. We won't.
I'm pretty sure we're not fast enough for that, because
that's what I was going to say.
You have all sorts of brilliant Wall Street folks who are learning about this stuff the second it comes out, the second that press release gets released, and then they sell or buy it. So if we're popping in 3 hours later being like, hey,
ooh, I found a press release, I'm so smart.
Yeah, we're already lost.
Yep, I agree.
The majority of the gains have been made.
Yeah.
Well,
do you want to jump into your first story?
Yes, let's do it. Very good. So, yes, the first one of the day here is going to be Hemologics. They have their phase 1. I thought this would be a really interesting story to look at. They're number 4 and number 5 on the Biotech Voyager leaderboard?
Number 5.
Number 5. Excellent. Yes.
That was their first patient.
A couple of interesting things here. It's an Australian company right off the bat, Jeff. And obviously the big signal being they've dosed their first patient. But I just wanted to take a little bit of time to just go through a little bit of their platform. So they're using a really interesting approach here, where they develop both the antibody and the CAR T immunotherapy for targeting 2 tumors exclusive antigens. So I thought this was kind of interesting. Not only are they developing, they're not just developing a chimeric antigen receptor. They develop the antibody and they develop the CAR T therapies. to go after this. They're going after 2 tumor exclusive antigens, a kappa myeloma antigen KMA, and then lambda myeloma antigen. And these are found on malignant B cells in multiple myeloma and related blood cancers, but absent from healthy immune cells. And so it's really interesting to see that they've kind of gone after this approach as opposed to kind of the standard inside of the Aussies so far, which has been, oh, let's all go in vivo CAR T.
Yeah, I'm wondering why, if these antigens are targetable, exclusively expressed on myeloma cells, why hasn't anybody made antibodies against them yet?
Yes, good question.
I always get a little bit skeptical when I see things like that. Let's go to their website and see if they mention that. Because I would be very curious to know, like, because what in my head I would immediately think they must have.
What? They did the thing.
What's the thing?
Fighting cancer is in our blood. Genes are in our DNA.
So, oh my gosh, come on, I can't get this. Yeah, I would imagine that they would have to have some kind of like antibody discovery thing. Proprietary monoclonal antibodies, kappa-mab and lambda-mab for patients with lambda-type multiple myeloma, and then kappa-type multiple myeloma. Yeah, that's very interesting. It's like a novel target antibody combination, and instead of making an antibody, they just use that as like the variable fragment as the CAR T cell targeting domain.
Yeah.
Okay, so they dosed their first patient.
They did
yesterday.
I believe it was yesterday,
or they announced it yesterday.
This is so first in human, obviously a big point, a major inflection point for a company like that. So
yeah, for sure. I'm wondering which one it is. If it's the lambda or the KM, whatever it was.
If you go up to the top, kappa. It says the KM CAR T cell.
Okay, yeah. Okay, so it's the kappa. Yeah. Interesting. And honestly, like I'm like a cancer guy, I guess. I've never heard of kappa and lambda type multiple myeloma. No clue what that seems to be something that's not super widely known. And this is very interesting. I don't really even know what they're really doing. Ben, we know somebody from Hemologics.
Do we?
Yeah, or at least he used to work there. I have one of their decks on my desktop, like right next to this screen. I'm looking at it right now. Yeah, interesting. Okay. So first patient dosed. Definitely keep an eye on this. Again, if anybody in the comments wants to talk to me about what kappa and lambda type multiple myeloma is, that would be helpful.
Yeah, because
I've never heard of it. Interesting. Did you look into it when you looked into the story?
I did not in a whole lot of detail. Full disclosure for the viewers at home, we have a 2.5-week-old baby, and old Ben hasn't been getting a whole lot of sleep recently.
Ben has other priorities on his list. Sweet.
But yeah, definitely cool. Kudos to them. Big milestone at a time when people are going back and forth about the benefits and attributes and drawbacks of CAR T cell therapy. This is a new one that's
true.
Along.
Lots of controversial news recently, huh?
Yes.
Cool.
But yeah. So yeah, next up, Arika, Jeff. Want to get into it?
Yeah. So, this story is technically about Arika. That's how I came across it. I saw the Arika press release, and they said, "Hey, guess what, guys? We won this competition." And I was like, and it was published in Nature Biotechnology, and I was like, "What is that? That sounds cool." Looked into it this morning. Now, fair warning, we're going to go through this kind of together. I looked into it a little bit, but the details are still, it's hard, right? You can't, I'm a biology guy, but I'm not Jesus. Okay. I I took about a half an hour to look through this. So, but what I can tell is that AI is being used to do all kinds of stuff in biology, in biotech. One of those things is it's being used for antibody design, antibody discovery, and antibody design. And the holy grail of AI-based antibody design would be giving the AI program an antigen target and saying, give me a high affinity antibody that is developable. Right. And have it just do it for you. So this competition it seems to be kind of whoever put it together gauging how close we are to that holy grail. Mm-hmm. So how close are we to giving an antigen and getting AI programs to give us a high affinity antibody that is highly developable?
Right.
And the answer was, drum roll.
I can grab that.
We are not even close to being close. Not even close.
Well, so make sure again. I can't see it.
You can't see it. What do you mean?
Just you're still in the Nature Biotechnology?
Yeah. So nothing changed.
Oh, okay.
I didn't. But the answer, from what I can gather from this, is AI is. It's good to do some things, but when it comes to just like, hey, here's this thing, go do thing.
Right.
It's just not there. So when it comes to the antibody generation, right? So there was what this thing was they published the results of this like competition, 29 companies, Aurica being one of them, 511 submitted antibodies. And they were given 3 challenges. They submitted their antibodies, and then those antibodies were subjected to 3 different challenges. And all these challenges were experimentally tested, right? So the first test was affinity maturation. So what they did was they gave the companies an antibody, a BRD binding antibody, and they said, use your platforms to make mutations in this antibody to increase the affinity. Right. So the company said, okay, so they took their antibody that they were given, they analyzed the sequence, and they said, okay, here's our antibodies that we made mutations to, we use the AI programs that we have to predict mutations that would increase affinity. Here's the thing. Here's our antibodies. They were tested, and this is where AI actually did kind of good, right? Aurica won. This is what they won. They won this part. So their antibody went. They found that it had 95 picomolar affinity. So they must have had some kind of threshold. And the more antibody you had to add to reach that binding threshold, the lower the affinity was, right? So like the less antibody you add to get to reach that binding affinity, the higher affinity. So the lower the number is, the better your affinity was. They got it down to 95 picomolar to reach some binding threshold. Now, that was about 2,000-fold improvement over the BRD antibody they were given. Right. So they could achieve the same binding affinity with 2,000-fold less concentration, which is really good for like therapeutics, right? Because you can lower the dose. But that's great. But then a company called Biogen took the potential mutations that you could have added to this antibody, and they just kind of like sat around a round table and discussed it for a couple of days and came to some kind of consensus as to which mutations should be and shouldn't be added. And they got it down to 540 picomolar. Just with like talking about it. Yeah. So it's like, okay, we have this super crazy AI powered antibody optimization program, and we can get it to 2,000 times more binding affinity. Right. And then we got a couple of scientists sitting around at lunch having a couple beers, and they got it down to 540. so it's like, okay, like, yeah, the AI did better and it worked, right? That's challenge number 1. And the takeaway here is that it's promising, but also like it's not a clear goal. Okay.
Right.
So number 2, affinity ranking within certain clusters. The goal was to achieve the highest affinity clones. This one, let me go real quick here. Challenge number 2. The teams, they gave the teams 3 HCDR3 sequence clusters and asked which sequences are going to be, okay, yes. So this was like predicting binding affinity. So essentially what they did was they gave a bunch of different antibodies to these people, a bunch of different antibody sequences to these people. And they said, we know which antibodies have higher affinity. We've already tested it experimentally. Which ones have lower affinity? But you go, don't do any lab experiments. Just use your AI prediction models, right? And you come back and you tell us which ones do your models predict have the highest affinity and the lowest affinity. And guess what? The AI sucked. It didn't do good at all. It said here that the authors had an experimental baseline showing that simply randomly picking antibodies from these pools gave the accurate results 40% of the time.
Oof.
The AI.
Oof.
So if you just randomly
choice.
randomly picking it got about 39% accuracy. The AI had an average of 10 to 14% accuracy.
Oh my goodness.
Yeah, and only the team from Washington University beat the random picking baseline. So the authors literally state that for most participants, using AI was actively counterproductive in this experiment. The AI actively made the results and the predictions worse. So this is pretty devastating for any company that's saying that our model can look at an antibody sequence and predict which ones have the best affinity, because they literally, not only can they not do that, but you're probably better off taking their answer and doing the opposite. If your antibody affinity prediction partner says, "Hey, antibody number 1 is going to have a higher affinity," pick number 2, because they're 90% of the time they're wrong, according to to these results.
So what you're saying is, at this point, AI is kind of like inverse Kramer. Are you familiar with this?
No, but I don't.
Oh,
I didn't watch
it up.
Seinfeld.
No. So it's like the Mad Money guy in the investing world.
Yes,
right.
And sell.
Yeah. He's historically been so terrible at calling which way the market is going to be, which way the market is going to go, that people have literally set up funds called inverse Kramer, where they just do the opposite of whatever he says.
Jeez.
And it's up like 60% a year. You gotta go. It's wild. Go
Wow.
look at it. You can
That's
look it
nuts.
up right now. So this is literally the same thing, but for antibody selection. It's the AI is the inverse Kramer of biotech.
Refers to taking the opposite position of Jim Cramer's stock recommendations, a contrarian strategy popularized online and formalized in products like the inverse Kramer tracking ETF, which launched in 2023 but closed in 2024 after losing about 15% of its margins. Okay, so it went down. That's funny though. So, but basically, yes, it's kind of like if my AI predicts this antibody has the highest affinity, it probably is not only not that one, but it's the one that it shows to have the lowest affinity that probably has the highest affinity. So, but that's just from these results. 29 different companies submitted 511 antibodies, and like they, so it isn't like AI sucks. Maybe it's just like these companies' AI wasn't good at this particular thing. That's the more modest claim.
Okay.
Okay, so then the third challenge was basically saying, here are 32,000 sequences and affinity measurements for 142 antibodies. Now generate new like CDR combinations that aren't in this data set. So it's generating a new antibody, right? From antibody sequences. And the results, and again, this is where I didn't truly fully understand this part. So the results were mixed. There were some genuinely spectacular molecules. The winning design hit 2.9 picomolar, and 24 out of 168 submissions were well below 100 picomolar. But simultaneously, okay, so yes, they said, here's a library of antibodies with affinity measurements, right? This now it makes more sense. Yes, okay. So they gave them a sequence. They gave them 32,000 antibody sequences, and 142 of those had affinity measurements. So you've got the antibody sequences, and a pretty decent number of those ones, what their affinity measurement is.
Right.
And then the challenge was create new antibodies that have lower affinity, or I guess higher affinity, right? This did okay. The winning design had the affinity was 2.9 picomolar, which is really low, and
Right.
24 of the 168 submissions had binding affinities below 100 picomolar, right? However, a third of all antibody submissions didn't bind at all, right? So the AI said this is going to bind really well. A third of the antibodies didn't even bind at all.
Oh,
Another
dear.
substantial fraction bound but failed developmentally. So, meaning, yeah, your antibody can bind, but there ain't no way we can make this thing at scale, right?
Right.
Only about half of all designs were both binders, not even good binders. Only half were able to bind and were considered developable under their benchmark criteria. So, even the headline 2.9 picomolar winner has an enormous asterisk next to it, because it's kind of like shooting, your gun at a barrel. Like, you're going to hit every once in a while, but the vast majority of the time it was just completely not working.
Okay.
So that was the results of this experiment. You can go and find the paper on Nature Biotechnology, and look at the results for yourself. But that seems to be the consensus of AI-based antibody generation. Promising, but we're not there yet. Unless there were companies who have better models that just like didn't participate in this.
I wonder that.
But
Are the best ones just being like,
we're
keep
holding
our
it.
cards pretty close to our chest?
Yeah,
not...
but you'd think that like this kind of publicized thing, you'd want to be in. It's kind of like I'm not going to go and compete at the Olympics because I don't want anybody to see how good I am.
Right.
It's like, right. I don't know. Maybe not entirely that, but I wouldn't see a reason to not participate.
Right. Makes sense.
Unless you didn't know about it.
Definitely. No, it's interesting. So it's not there yet, especially when it comes to designing, right? Like very large, very complex proteins, and not having all the different data points that are going to pop into that as well. In terms of just how vastly complicated the inner working dynamics of a cell are. Good luck at this point making something in silico unless you've got all of those thousands of different interactions properly mapped. It's going to be challenging, and I think this paper kind of shows that.
Yeah, one of my big theses of like the biotechnology space compared to other technology spaces is other technology like energy, cybersecurity, software. You're solving problems that humans created, quote unquote, right? Like the energy crisis or energy is you're solving a human problem. And it's a vast oversimplification, but hopefully the point is kind of driven home. Software, you're building within the realm of like the hardware that we've got. It's a human designed problem. Automobiles, right? When it comes to biology, biotech, we aren't dealing with it's a different category of problem and a different category of solution design. We aren't dealing with cars or computers or like how to make better turbines for charging up our batteries, right? This is like a problem that there's literally an infinite number of unknowns relative to the number of knowns.
Yeah.
And we're dealing with things that are ancient and biological and like natural, and we can't really expect to apply the same principles of like tech solution to biotech and see the same results as we do in like, software, for example.
Yeah. At least not yet, but that might be a conversation for another day. That being said, Dan has joined the green room, and so we'll be bringing him on in just a second here. So, ladies and gentlemen, we will be speaking with the CSO of Camp4 Therapeutics. Hopefully, he can turn his camera on before we bring him in. Perfect.
There he is.
Excellent. So, yes, ladies and gentlemen, if you have any questions for Dan, once we bring him on, by all means, feel free to make them known, and we'll get to them as soon as possible. But yes, with no further ado, let's bring him onto the show. Dan, welcome to the show.
Thanks for having me. Great to be on. Appreciate it.
Well, we appreciate your time. So yes, right from the get-go, obviously we'd love to learn a little bit more about what you're working on over at Camp 4. We're big fans of the approach that you guys are taking. But I guess could we just get a little bit of a background on you, like what kind of brought you to Camp 4? What's your educational background?
Yeah, sure. So I'm the CSO of Camp 4. I've been here about 3 years or so. And, my background is really in drug discovery, largely focused in the neurological space. So prior to joining Camp 4, I was at Pfizer for a little bit in their rare disease group, doing some looking at rare neurological disease. But before that, I was at a company called Humanity Therapeutics. And this was a
Mhm.
small molecule company focused on neurodegenerative diseases such as Parkinson's and ALS. And this was actually work that was stemming from work that myself and others had done at the Whitehead Institute, MIT, during my postdoc. So Susan Lindquist was our founding scientist, and I was a second employee, and we really sort of built this company and brought some small molecules forward for neurodegenerative disease. So that was my postdoc at Whitehead and MIT. I did my graduate work in molecular and cell biology, sort of very basic understandings of gene expression at Brandeis University. So that's sort of my path here, and I've had a really fortunate opportunity of working with some great leaders in the field and getting exposure to drug discovery, and now here at Camp 4 overseeing our platform, which is happy to get into that in a moment, as well as trying to advance our early pipeline. And we're really excited about where we've gone from trying to establish a platform and actually get drugs to the clinic, which we're excited about. We're taking some steps later this year.
Excellent, fantastic. So yes, want to get into that in just a few minutes, but I guess just to kind of set the stage here for anyone who may not be as familiar, can you kind of just give us a little bit of an overview, like what are ASOs and why ASOs?
Right. So ASOs, or antisense oligonucleotides, are single-stranded nucleic acids essentially, and they're able to hybridize or bind to specific RNA sequences. So, as everyone knows, genes are transcribed into mRNA. The biology is very complex, and you can actually target these target RNAs and have a multitude of different effects on RNA biology. Ultimately, people have most often used antisense oligonucleotides to decrease gene expression, to actually knock genes down, and you can do so in a very precise way. And this is there's a lot of work over a number of different targets of actually taking some disease protein of interest, for example, and decreasing the levels of that, and where a decrease in that expression could have a clinical benefit across many different types of diseases. So I think the advantages are it's very sort of precise, precision medicine. You can go after targets that might not be amenable to small molecules, right? So,
Yeah.
small molecules have certain things they're very good at doing in terms of inhibiting certain types of enzymes, sort of agonist antagonist biology. But with an oligonucleotide, you can theoretically go after any RNA target, and I think that really opens up the therapeutic space and types of indications you can go after. And then it's really sort of matching the mechanism of those oligonucleotides, which we can talk about, where we're interested in upregulation, matching that to what you're trying to achieve in your therapeutic indication.
So I have a question. Thank you for the explanation. It's basically like it's kind of like siRNAs in lab, kind of, right?
Yeah.
I've always wondered. I'm a cancer guy, right?
Mhm.
I've always wondered, and you mentioned that they're really good for targets that are traditionally undruggable with small molecules or
Mhm.
antibodies, right? Why haven't we seen this in oncology a whole lot? Maybe we do. Maybe I'm just ignorant to it.
Yeah, I mean, there's definitely some work in that space. There's also, just to the point of maybe just clarify on antisense oligonucleotides versus siRNA. They're essentially an antisense oligonucleotide is a single-stranded, um, can again hybridize directly with RNA and have some impacts. In terms of siRNA, you're looking at direct downregulation of the target through it's loaded with a RISC complex and sort of
Yeah.
finds a target, results in degradation. So ultimately, for knocking down, both those mechanisms work out quite well. I think in the oncology space, there definitely is less of that in the oligonucleotides. I would say there's a lot of the work with oligos and siRNAs is in the field, looking at in the lipid metabolism space, for hypercholesterolemia, you look at, reducing cholesterol, things like that. I think there is work in the oncology space. I think sometimes it depends on what's the amount of decreased expression you might need to have an effect in a cancer cell where you really might
Okay.
completely kill those. I think
Sure.
often with knocking things down, it's not knocking things out.
Out.
It might be the situation where an oligo, you can reduce something 50%, 90, maybe up to 90%.
Mm.
Is that 10% remaining? Is that enough to allow cancer cells to sort of survive, right? So with a small molecule, you might be able to, if you have the right target, you might be able to get a higher degree of pharmacology. So I think, one nice thing about oligonucleotides in general, whether it's antisense oligos or siRNAs, is they're titratable, right? So you can alter your dose, to really achieve the level of target engagement to have the effect that you want. And small molecules are as well. Uh, but I think, especially on the knocking down side, again, it's sort of down, not out in most cases. And I think that might be part of the reason it's a bit more challenging in the oncology space.
That makes sense. Yeah.
Love it. Awesome. So I guess, yeah, to dive a little bit more into your platform, just to kind of set the stage there a little bit further. When you're kind of looking at the problems that you guys are trying to solve across these different therapeutic indications that you're going after, why have other approaches failed here? Like what's the unmet need here that other approaches just haven't been able to do a good job in so far?
Right. So I think, really what we're trying to achieve is gene upregulation, right? So I think, there's a couple approaches to increasing the expression of a gene. The class of diseases that we're going after right now, especially in the central nervous system, are their haploinsufficiencies. And what that means is that, one allele of the gene has a mutation that results in, loss of expression or loss of function of that gene. So you essentially have 50% of the amount of protein that you should have for
Ah.
that gene, right? So that's what a haploinsufficient disorder is. Um, and there's a couple approaches here, in terms of like increasing expression. That's really difficult to do with a small molecule. That's not really what they're built to do. So you then want to think about a genetic strategy. And I think that the two main areas there are oligonucleotides. And I think, again, while oligos started out with downregulation, where we and others are trying to upregulate, or gene therapy, right, where you have, a gene that's housed in a capsid and AAV, and you're trying to introduce that and actually express exogenously and replace that gene. So I think you need a genetic approach for these disorders. That maybe there's a thought you could think of editing in some cases as well.
Mhm.
But it's really, I think oligonucleotides are poised to have an approach here, to have an impact here. So then you just got to think about what's the mechanism that would allow you to increase expression. And again,
Right.
that decreasing expression is easy, but fortunately for us, but easy. I mean, it's relatively easy. Um,
Sure.
uh, Upregulation really starts to tap into some of the very complex aspects of mRNA metabolism and the
Yeah.
fate of an mRNA, right? So there's a couple different mechanisms that people have used to explore upregulation. And this includes splice switching oligos, and I can get into some
Mm.
more detail there. And what we're trying to do is to target really a fundamental class of RNAs that control gene expression called regulatory RNAs. And by targeting these with antisense oligonucleotides, we're able to effectively increase the expression of any gene that we want. So if there's a disorder that's a haploinsufficiency, and we therapeutically, you
Mm.
want to bring levels back up to normal. Our approach is able to do that with what we've looked at so far, and that's essentially what we're trying to do in the clinic.
Okay. Any gene that you want implies that every gene is regulated with post-transcriptional RNA
Mhm.
like regulation like this.
Yeah.
Is that true?
Um, it's always a little dangerous to say 100%, right?
Yeah.
But I'll say most. So we actually,
Okay.
our platform, and just to get into this a little bit. So first, maybe just a
Yes.
little more on the biology, right? So these regulatory RNAs, they're non-coding RNAs.
Mhm.
They're actually expressed from the genetic regulatory elements of a gene, right? And this includes promoters. So typically, right, a promoter is where your transcription factors will land and help drive the expression of the messenger RNA. It turns out that promoters are actually transcribed in the opposite direction, on the opposite strand. So you get these antisense transcripts. We call these promoter-associated RNAs. And then enhancers, which are genetic regulatory elements that are typically more distal from promoters, and they actually will come into contact with promoters and help regulate gene expression. They're transcribed as well. So you get a bunch of RNA that's being made in addition to your gene's mRNA. And what we found, and this is work supported by one of our founders, Rick Young at MIT as well, who is very interested in transcriptional regulation, is that we can target. One is that these regulatory RNAs specifically control the gene that they're nearby for the most part, and then we can target these with oligonucleotides to essentially change the interaction of transcription factors with the gene of interest. So the first thing that we do of our platform is actually to map the regulatory landscape of the cell type, right? And we can do this in cells in a dish, in like iPS-derived neurons or hepatocytes or in different cell type we want, as well as tissue. So looking directly in liver or heart or brain, for example. And when we map these regulatory elements, it's a genome-wide map, and we see, from a cell type, we've observed, 130,000, for example, different regulatory regions within a genome, right? And that's controlling maybe 15,000 different genes that are being expressed. And it turns out, most of these are actually actively being transcribed, and that transcription is contributing to regulation of that gene. So it seems that the transcription of these regulatory RNAs from promoters and enhancers is, we'll say, a near universal feature of genes, right? And in most cases that we've looked at, we can identify these regulatory RNAs, and then when we target them with an oligo, we can detect increases in gene expression. So, is it 100%? Maybe not, but it certainly, in the most cases, we can find these regulatory RNAs and have it be able to intervene and increase transcription. And what's important is the data that we've generated so far, really suggests that this effect is specific, right? So if targeting these regulatory RNAs was having an effect on dozens of genes, that's not great, right? Maybe we're increasing expression of our gene of interest, but also a bunch of other things, and we don't really want to do that. But it really seems that these RNAs seem to be specific for the gene that they're close by to, so we can have a pretty specific effect on our gene of interest. And our lead program in a SYNGAP1 related disorder, it's a neurodevelopmental disorder. We're specifically increasing the expression of that gene alone that's mutated in patients with that disease.
Okay.
Okay, my brain is it's coming together in my brain, but forgive me if
let me see if I can get you there.
Forgive me if this comes out kind of clunky. Because it would imply a few things. This is new to me. I guess I had no idea that there was near universal regulatory RNA based gene expression regulation in our cells. I didn't know that. I figured that it was a very isolated thing with certain genes, and it was very deliberate kind of in terms of transcriptional regulation. But it would also imply that just kind of at homeostasis, the transcriptional process is wildly inefficient. Like it's making transcripts that it's just going to degrade all the time. It's kind of like a premise of your technology would be that we can repress the repression and get a significant increase in the expression. Right?
Right. Yeah. No, that's good point. So I think, it's been known for quite some time that there's, in addition to mRNAs that are being transcribed from a gene, there's a lot of other transcription. And I think probably initial thoughts were this is just junk, right? You just you're getting this spurious transcription all over the place, and it's not meaningful. You've probably heard the term sort of dark genome, looking in place non-genic regions for either, regions of the DNA that have an impact on disease, or areas that are actually expressed or actually transcribed. So I think it took some time. I think sort of the first observation was, oh, that's interesting. There's RNAs that are being made from all these regulatory regions. Is that important, or is it just sort of an effect? Is it just happening as a consequence of there being a bunch of transcription machinery there? But it really does seem to be important. Like it seems to be, playing a role in the regulation of transcription. That when you have RNAs being made from an enhancer, they're not just wasting energy by doing that. They actually are playing a role in helping regulate transcription. And our mechanism for what is happening is this is all happening in a very kind of dense, chaotic, three-dimensional space, right? Like
Yeah.
often we look at things kind of drawn out on a paper, but really what we believe is that these promoters and enhancers are actually coming together, even in what you've probably heard called a condensate, which is essentially like an accumulation of proteins, and they kind of have specific set of properties. And these RNAs seem to what they seem to be doing is actually interacting with transcription factors, right?
Sure.
And transcription factors are what help recruit the transcriptional machinery to the promoter. So you can think of these RNAs as they're being made locally, and then they're kind of binding to transcription factors, and they're kind of like, we think of it as like a kinetic trap, like they're preventing the transcription factors from like floating away, far away from the gene. So by being expressed, they're kind of hanging on, in a way to the transcription factors and helping regulate and sort of influencing that sort of dynamics of transcription factors on the DNA or off the DNA. So that's sort of our model, and that's, there's work sort of supporting that, academically, but ultimately it seems that it's important, right? If we were targeting these regulatory RNAs and nothing was happening, you could say, okay, well, maybe they're junk or something, but they really do seem to be tied to the regulation of transcription.
That's so wild. This is a little bit random, but I studied like fusion biology in grad school. We were doing Ewing sarcoma stuff, and we went down a little bit of a rabbit trail for a couple months studying the effects of the reciprocal transcript of the fusion, because both chromosome breaks get fused, and the one creates the fusion protein. The other one still transcribes sometimes, and that transcript can actually lead to the promotion of the pathology. And it's just like things that you would never think of. I would never, ever, ever have thought of like topologically associated domain transcription leading to like transcription factor binding, and it creates like glues and nets, and that's incredibly complicated. And whenever I learn these new things in biology, I'm just like, how has it's been happening since the dawn of transcription, probably. And we're just kind of now becoming aware of it. And it's like, what is going on down there, man?
Yeah. I think as they say, right, reading textbooks and then X number of years later, everything is different, right? I think that's, everything is just more complex, right, than you initially thought. And you make these discoveries, you wonder if they're real or if they're meaningful. And, sometimes it takes time to realize they're meaningful. And I think for us, we've spent the time to establish this platform, and, there's always this, challenge of understanding the biology, but also trying to set that up to help discover drugs and develop drugs, because that's ultimately what we're trying to do, is to actually get molecules that we can put in patients and hopefully see that their symptoms improve. So, I think that's one of the great things about Camp4 and sort of a platform and sort of science first company is it's exciting to continue pushing areas of the boundary. I mean, I gave you some models. I can't tell you exactly that we know everything that's happening, but at the end of the day, we know we can increase transcription, and we think we're understanding, starting to understand kind of what's happening there. So no, I think that's really a compelling place for us to be, is sort of, again, pushing the science, while we're trying to advance a platform or advance a pipeline and get into patients, and trying to get better about it and learn as we go to make sure we're more efficient and can, bring more molecules forward towards development.
Yeah, like awesome. This right here, hang on real quick. This right here, like what's going on here?
Yeah, what's going on? Yeah.
Like this is crazy to me. I can't even tell you. So is the gray area like this is what are these egg things doing here?
Well, yeah. So I think so this is a slide from our deck, and when we talk about our platform, when I was talking about mapping the regulatory regions throughout the genome, how are we doing that, right? And the key thing that we have to do is generate really a number of data sets that are essentially looking at the expression, looking at the chromatin state across the entire genome. So what's shown here is essentially some RNA polymerases there that's making a messenger RNA in blue, and then you can see in orange, these are the regulatory RNAs. So the one there that you're pointing at towards the right side, that's the antisense promoter associated RNA. Okay. And then the one towards the left, that's binding to that green sequence. That's the enhancer associated RNA, right? And these are just all cartoons. But the things that we look at, like when we're actually doing the mapping, is looking at chromatin accessibility, right? So when an area of the genome is transcribed, the histones kind of move out a little bit. So you'll actually have accessible DNA.
Yeah.
You're looking at histone modifications. So, histones, as you presumably know, are highly modified. They're acetylated or methylated and different patterns, and those patterns on the histone tails sort of recruit various proteins. And there's certain ones that signal active or repressive chromatin. There's this DNA looping I sort of mentioned, right? That enhancers come into proximity with promoters. And so we have a way using what's called Hi-C to actually look at that. So we basically generate about a dozen or so different sort of genome-wide data sets that are really trying to look at the regions across the genome that are actively transcribed. And then we have go ahead.
Well, I tried to do some Hi-C sequencing in grad school. We had some CTCF hypotheses. We couldn't get it to work. It's pretty complicated, the assay.
It's not straightforward. And I think that's where, having a founder like Rick and a postdoc of his, Ala Segova, who's the head of our platform, she's really helped to bring that here. And it's definitely something that takes a good set of hands on the team to do. It's probably one of the less straightforward assays. As far as I understand, I've never tried to do it myself. But as they say, I think it is quite complex. So I think it's, once we get all this data, right, we have sort of a machine learning algorithm type thing that we've generated to help basically identify and score the regions. And then just sort of to continue to finalize sort of what the platform is, right? This points to the region of the DNA that we think is expressing a regulatory RNA. But we then actually have to confirm that these regions are in fact making an RNA, right? So what we do is we will have a regulatory region, and then we'll tile the entire region with oligonucleotides, antisense to the predicted RNA, and pull them down. So actually affinity purify the predicted regulatory RNAs, and then do long read sequencing to actually capture the full length of these RNAs. So first, the mapping points us to the DNA, then we confirm the RNA, and then we just get to screening. Once we have those RNAs, we can start screening for oligos, looking for those that increase expression, and that's kind of more typical drug discovery and development once we go there. But that sort of all this mapping and understanding of transcription is really a very kind of unique thing, I think, to what we're doing, and very exciting.
Yeah, and you're mainly doing it in haplo-insufficient disorders.
That's where we're starting. We initially had done some work in a liver target called CPS1 for urea cycle disorder, but we really felt that wasn't exactly a haploinsufficiency, but we were generating some data in some CNS indications with very clear haploinsufficient mutations, and SYNGAP1 was part of this, and we really were excited about the data, and it really was amenable essentially to the effect on transcription that we're having. So maybe just to point this out, right, we're not getting 5-10-fold increases in expression. We're getting like twofold, right? So that might be a bit of a limitation, but it's also really it points us to which therapeutic areas could be beneficial with our or fit our platform, in terms of like where a modest increase could have a pretty significant benefit. But it's also nice that, from a safety perspective, I think there could be concern about increasing the expression of a gene tenfold, right? I don't think from a safety kind of tox and development perspective, that's not great either. So really nicely, the effect size we achieve is really nicely suited to haploinsufficiencies. And, SYNGAP is really interesting. Again, it's a neurodevelopmental disorder. These patients have developmental delays, intellectual disability, seizures, sleep disturbance, very significant behavioral disturbances, and it's related sort of in the same class of disorder as Dravet syndrome, which Stoke Therapeutics has been working on, and they have a splice switching oligonucleotide to increase gene expression. That might be a mechanism that people have heard about, where you have the inclusion of an exon in a pre-mRNA that results in its degradation. If you can prevent that from happening, you can theoretically increase the amount of mRNA that's making protein. So that, there's a lot of learnings that they've had there, and they're having some really, really nice signal in their clinical studies. So that's an approach of upregulation. But SYNGAP is related to that, and there's a number of diseases in this space of what we call developmental epileptic encephalopathies, where they're haploinsufficient. They have a related sort of set of phenotypes in terms of neurodevelopmental and seizure endpoints. So it's really an interesting area for us to be in with our mechanism, which is sort of this kind of physiological bump in gene expression.
I would love to hear you say that 5 times fast.
I would. I
Or
prefer not to.
or hear Ben say it one time slowly.
Let's go with that. But yeah, no, there, you can't always control the words, but yeah, it's it's been a great experience. We really over the last year have talked a lot about our data for that program, which we can show, increasing in patient iPS-derived neurons. We have a humanized mouse model where we see nice increases and increasing expression reverses phenotypes. We can administer to monkeys and with intrathecal administration, which will be how we'll dose in the clinic. And we can see nice, good biodistribution, increases of expression. And now really what we've been working on in the last, or what we've been working on is how do we get to the clinic? And, we're very excited. We recently announced that, we had regulatory approval in a couple of jurisdictions in Australia and Argentina, and we have a couple other submissions that are in review. So, we really are anticipating starting our phase 1/2 clinical study in patients with SIGAP1 disorder, by the end of the year, by targeting these non-coding regulatory RNAs that are sort of an emerging, sort of an exciting class of RNA. So it's been great for us. And behind that, we continue to think about where else what other targets make sense for us. Again, I sort of mentioned there's a number in this space, and we're trying to build on what we've learned, with SYNGAP and apply that to other areas as well. So I think we're in a really, really nice place to continue doing exciting science and hopefully make a meaningful impact in patients' lives.
Excellent. So I know we've only got about 3 minutes of your time left, and we want to be super respectful of that. So two things I just want to cover quickly. So first of all, so if I'm understanding correctly, just in terms of broad strokes again, when people think normally of a gene therapy, you kind of tend to think of a disease where the target protein or the target gene is mutated in some sort of way and it's no longer functional. You're providing the new copy. But there's this basically an entirely different section of disease where essentially the gene itself isn't mutated so much as it's just not getting expressed properly. It's not getting expressed enough. And so ASOs really have this potential to come in and address these thousands of different diseases where that is the case. The person just needs the gene in particular to get upregulated a bit.
Right. Yeah, that's correct. So just one thing about our approach is you're going to be increasing expression from both the wild type allele that's fully healthy and functional, and in many cases, the mutant allele that is resulting in a haploinsufficiency. It's often a mutation called a nonsense mutation or a frame shift where that RNA is actually just being degraded and you're not making any functional protein. Sometimes you'll have missense mutations or amino acid substitutions where you have full loss of function or the protein is no longer stable. So by targeting the regulatory RNAs, we are increasing both, but you're essentially increasing the healthy one, and then you're increasing one that's basically just being degraded anyway. So that's really, for example, in SYNGAP1 related disorder, about 80% or so of patients have either nonsense or frame shift mutations where they have a mutation, a premature stop codon, that RNA is just being degraded. They're never even making SYNGAP protein from that dysfunctional allele. So we are increasing both, but the mutant one, there's really no impact of that.
Awesome. Yeah. Perfect. Okay. And then last question. This is a question we try to ask all the guests that come on the show. It can be a conglomeration of everything we've talked about so far. It can be something else entirely. And it's just this idea of what's slept on in biotech right now that's going to be a big deal in a couple of years.
Yeah. That's an interesting question. I think, in the space that we're in, and I'm not sure if it's entirely, slept on, but it's just something that's really growing. And I think there's a lot of interest is how to deliver oligonucleotides, be it ASOs or siRNAs, to the CNS without having to do an intrathecal administration, right? Yeah. So, we're going after a disease that's, very high unmet need, very severe in nature, and the idea of doing maybe like, every 3 or 4 months doing an intrathecal lumbar puncture, it's okay, but it's a rare disease as well. So for larger diseases or maybe less severe diseases, intrathecal might not be the best. So there's a lot of effort using blood-brain barrier shuttles right now, or using, the idea is hooking up your oligo to an antibody that's targeting a receptor to get across the blood-brain barrier so that you could do IV or sub-Q dosing of an oligonucleotide, get it into the brain. And and so it's a much different experience, right? Having an IT and dosing versus, IV dosing. there's a
Yeah.
lot of work there. There's many different ways that companies are looking into this, and there's been a couple of successes, but I'd say the field as a whole is on the earlier side. But it's something, I was at a conference the other week, and it's just such a predominant and emerging area in the CNS oligonucleotide field, where it's definitely the direction that people are going, and some interesting, exciting data coming out, but also a little bit more kind of proof of concept, as well as technical things need to be worked out. But I think that's something that we should really, in the CNS oligo field, you'll see evolving in years to come.
Excellent.
Yeah, one of the biggest patterns we've seen in the Biotech Voyager is CNS-related disorder therapeutics, and it's got to be with the blood-brain barrier penetration technologies across the board: antibodies, small molecules, AAVs, all over the place.
Yeah, definitely a big area.
Yeah,
excellent. Dan, really appreciate your time. Thank you so much for coming on the show, and yeah, we look forward to watching what you guys do next. Thanks so much for your time.
All right, thank you. It's been a blast. Thank you.
Thanks, Dan.
Right on, Jeff. That's wild.
Yeah, I did not a lot of work on it, but we did do some like topological associated domains, transcription regulation. It's crazy, dude. Like he said, when you're in elementary school or probably high school, maybe college, you learn about transcription and you see DNA and then RNA polymerase complex binds, and then you RNA. It's turned into a protein, and that's
it's so clean.
Reality is just like this.
Yeah.
All the time, and things bind and they don't bind, and there's all kinds of stuff, and it's like, and even he mentioned, like, I won't claim to know everything that's going on down there. It's like, we don't know anything. Like, we can, if we had a pie graph of all the things that we know that are happening in the cell, and we had a sliver of like what we know, it'd be like infinitesimally small.
Yeah.
Basically by principle, right? Like we don't even know what we don't know, right? So it's crazy. And again, like that picture on there with like the regulatory RNAs binding to transcription factors and regulatory factors, and it's like, what? That's a completely different level of regulation that I never even heard of.
Yeah, you brought that up, and I was just, as someone who has been now in cell biology for over a decade, I was floored once again at just how complex this thing is. Like, our scientific textbooks, we think they're getting more and more complicated as we go into university, and then they're just not.
Well, and the thing is like it's the classic principle of like, the more that you learn, the more that you realize that you don't know.
Yeah.
So you're like chasing this train that's getting faster and faster and faster. Yeah, I mean, I've known of like regulatory RNAs that are like RNAs that bind to other RNAs, and they can, regulate gene expression that way. I've never heard of RNAs that bind to and tether transcription factors to transcription complexes. That's like completely new. Learned that on August 27th, 2026. So yeah, and I've been in this thing for 12 years.
That makes you wonder, like, how does that start to impact like gene therapies and other types of mRNA?
Well, and they aren't even touching that stuff. He's talking about using an ASO to downregulate the presence of a regulatory RNA that directly downregulates the expression of some like therapeutically relevant gene. Right. They're not even talking about like using their technology to identify and therapeutically target the regulatory RNAs that bind to transcription factors and stuff. Like, what they're doing is still relatively simple, still very complicated. But again, like he said, we do all these different high C experiments, and we'll get these like topologically associated domain data and transcriptional data, and we get these candidates of areas where we might be getting the regulatory RNA expression. We'll get the, then they do all kinds of like cis sequencing, which is insane. I had some friends who did that in college. It's crazy stuff. But then they're doing these screens. They'll get these candidates, do the screens, like which of these ASOs increase the expression of our gene of interest, right? At that point, the mechanism doesn't even matter, right? Yeah. Like, and it could be acting in all kinds of different ways that wouldn't even really matter. It's just nuts, man. It's really crazy. That's good for them. No wonder they've raised so much money. they raised, I think that the signal recently was that they raised like $100 million, like it's a public offering or whatever. But they're swimming in it, and it's like, well, yeah, they're creating their own data sets using like high C. High C is not simple.
Yeah.
We can go on for hours on that kind of stuff, but I'll spare you my nerdiness.
Right on. Well, yes, shout out to Dan for coming on. That was super cool
Yeah,
to learn
very
about.
cool.
Excellent. All right, Jeff, we've got a couple more signals to get through for today before the show wraps up. So Socio is our next one here. If you're able to bring that up.
Yeah.
Socio Biotech.
Oh, they're in
ADCs.
Prague. Nice.
Prague, right on, European style. So this one's pretty interesting. So again, they're developing ADCs for difficult to treat solid tumors, but in particular, they have a pretty cool section of best in class assets, and they just got an orphan drug designation. So that's the big signal today.
Yep.
ADCs, kind of like the quiet workhorse of the industry, doing just a whole lot of good work, and making a lot of different people a lot of money. But really cool to see something like this that's just kind of considered the workhorse to be getting breakthrough designation.
Yeah, and they're doing it. So this was for breakthrough
orphan drug designation. Sorry.
So fast track, FDA grants fast track designation to Socio's SOT 106 ADC for soft tissue sarcoma treatment. Soft tissue sarcomas. I studied Ewing sarcoma as like my PhD project.
Right.
It's a soft tissue sarcoma, technically speaking, even though it's a bone cancer, right? It arises from the soft tissue around the bone. They're rare.
Right.
But they also primarily target like pediatric. They're normally pediatric. So this is kids we're talking about. Right. And I'm wondering like, yeah, what is the target? What is the because soft tissue sarcomas is like saying like carcinoma. It's like every solid tumor basically is a carcinoma, essentially. Right. So like which ones? But yeah, super interesting. What do you know about their technology? It's an ADC, duh.
I don't know a whole lot. Again,
targeting LRRC15, okay. For sarcoma and other solid tumors. Interesting.
Yeah. So they received both orphan drug designation for osteosarcoma in June.
And
then they got fast track designation just now for soft tissue sarcoma. So
again,
they've been on a little bit of a roll.
So osteosarcoma is like a bone sarcoma. That's right. Which is this here. Are they both LRRC15? I'm wondering if LRRC15 is like some kind of like preferentially expressed on sarcoma surfaces. Yeah. But yeah, really, again, they're relatively rare. Osteosarcoma is like one of the more common sarcomas. Right. But they're usually pediatric in nature. Good to see. Good. Mm-hmm. Oh, they're loaded too. Dang. Okay. Interesting. They're really loaded. Yeah. Huh. They're already in phase 2. They've got all kinds of stuff going on. They got CAR Ts in the clinic.
Socio, good for you. Let's go.
Yeah, lots of CAR Ts in the clinic. Actually, looks like. Huh. Cool. Nice. Cool. Okay, really quickly, I want to go into T-Rex Bio.
Absolutely.
So T-Rex Bio, as you can possibly surmise from their name, are doing Treg focused therapeutics. I'm almost positive, before we scroll down here, I'm not sure. In fact, I'm pretty sure that they're not doing like Treg cell therapies. Correct. It's like small molecules that target Tregs.
That's right.
But recently, they published clinical data. So let's take a look. So yes, so it's a TNF receptor 2 agonist, which TNF receptor is probably a activating, like it's an activating receptor for the Tregs. So I'm going to guess that this is for autoimmune disorders that they're doing this for. Let's see. So focused on tissue Treg biology for the discovery and development of immunoregulatory medicines, today announced unblinded top-line results for the first-in-human phase 1A portion of its ongoing phase 1A, phase 1B clinical trial for TRB-061 across the single and multiple ascending dose cohorts. Generally well tolerated, showed consistent pharmacokinetics. In addition, following single and multiple dose administration of TRB-061, selective reproducible activation and expansion of exploratory biomarkers inducing regulatory T cells was observed, supporting proof of mechanism and dose selection for the ongoing phase 1B portion in patients with moderate to severe atopic dermatitis. Yep. So they found good pharmacokinetics. It's safe at the the therapeutically relevant dose, and they found biomarkers to suggest an expansion and activation of Tregs. So all good news in atopic dermatitis, which is like inflammatory, right?
Yeah,
that's what that is. Yeah. So what they're doing is they have TNF receptor agonist small molecules that will preferentially bind to and activate Tregs to cause immunosuppression and tamp down on the pathological effects of atopic dermatitis and other inflammatory diseases.
Nice.
Yep. And that's, you'll probably see more now that they've got good like the top line data from phase 1. You'll probably see, let's see where they're at right here. An oversubscribed round earlier this year. Oh, they're loaded. Okay. You'll probably see like a Series C come through soon or a Series D come through soon to push this thing further into the clinic. That's probably the next signal you'll see. Or like first patient dose in their phase 1B cohort.
Absolutely.
Mm-hmm.
Love it.
Good for them. All right,
T-Rex.
last Revere.
Last Revere. Jeff, this is
All Revere.
I'm doing this almost for the meme.
Okay.
This one, I'll tell you why. This one, you got to take a look at their the platform they're using.
Okay.
So they call it.
Nice.
So, game recognizes game. But that being said, it is also just a really cool approach. So they are using small molecules to go after RNA splice modulators. And so I feel like that's something we need to explore a little bit further on the Biotech Voyager as well, right? Is to what extent can you start using small molecules, not just to like influence like signaling pathways inside of the body or inside of particular cell types, but can you also start using them to influence genetic sequences, which is, I think, pretty interesting.
Yeah, we did this a couple of weeks ago. I'm trying to remember what the company was called. Oh my goodness, I can't remember it. They're doing like small molecules that affect splicing Dan was talking about the inclusion of poisonous exons that cause the degradation of certain RNAs. You can actually influence splicing to either keep those in to cause the degradation of harmful RNA transcripts, or you can help to affect splicing to splice those out to increase the expression of potentially beneficial therapeutic transcripts. But I forget now what the company was called. Dang it. I want to say Pretzel, but it wasn't Pretzel. I forget. No? I forget. It doesn't matter. It does matter. It's gonna bother me, but we'll figure it out later. But yeah, I agree. It's super cool. That's also one of the things where you think of like genetic medicines or whatever as like nucleic acid-based therapeutics, and it doesn't have to be. Scribe Therapeutics is an example, although that's probably not a good example. But yeah, small molecules to affect splicing, and the thing too is like the nerd in me just gets like kind of giddy when it comes to that kind of stuff, because it's more than just like we make molecule to bind to enzyme no work no more, right? It's like, although it technically is kind of that, but like the downstream consequences, like the splicing enzyme not working anymore isn't really the therapeutic intent, right? The therapeutic intent would be that splicing agent is no longer working, and now splicing is affected, and you get a change in transcription, and then that induces some kind of therapeutic effect. That's cooler to me, and it's funner to think about than just like a KRAS inhibitor. No offense to the KRAS, no offense to RevMeds, which RevMeds just had their KRAS inhibitor FDA approved yesterday.
I saw that. We should have covered that as a signal today, but Again,
they're
Jeff,
obviously not on the
that's
Voyager, but
yeah, that's a later stage signal. So
yeah.
we'll
So
respect the game.
it's still gonna bother me what that company was called, but they've got to still be on the leaderboard here. But yeah, anyways, I'm still giddy over talking to Camp 4. That was super cool.
Yeah, wild how much we just don't know about biology.
Yeah, it's crazy. It is
Yes.
crazy.
So there
Hot
might
take
be
from Jeff.
Yes, so there might be a recurring series in the coming months on the Biotech Voyager where we talk to experts about specific modalities in a much more meaningful way like this. So stay tuned for that, folks. It's always good to bring on the executives of the people who are building these medicines, but we also might have the chance to do like a deep dive with someone. So stay tuned.
It could have been Autobahn.
Could it have been? Small molecules. Deep penetration of the brain.
No, dang it. That wasn't it. Now I'm way at the top again. Well, I gave it a shot.
Nice.
Anyways.
Well, Jeff,
what do you say?
We can probably leave it there.
Mm-hmm.
Ladies and gentlemen, thank you for coming to the Biotech Voyager. As always, we appreciate your time. We appreciate your patience as well as Ben tries to deal with having 3 kids in the house and also running a live show. So I'm slowly adapting to that.
I have forgiven you, Ben.
I appreciate it. Excellent. All right, folks, we'll probably leave it there, but thank you so much for coming, and we'll see you next time. Peace.